Or
You got promoted, Brian.
yes, exactly what I needed. Well, welcome everybody. Good morning, good afternoon, wherever you are. Good evening in some cases. Very happy to have everyone join us today. Very happy to have Dr. Ida Deichaite from UC San Diego join us. Ida is an assistant adjunct professor, radiation medicine and applied science at UC San Diego. And she is doing, she's a virologist, a microbiologist by training, and she's been doing significant research on tumor necrosis factor, TNF, and its relationship to immune response in prostate cancer. Beyond that, she is the director of industry relations at Moores Cancer Center. And Ida and I go back to, I think, 2017, when she boldly asked me to present some harebrained idea that I had about how medicine should be approached. And so we've been fast friends ever since then. And she and her teams have been very helpful in terms of looking at my genomic and multi-omic data to identify potential treatment options. So she's a friend, amazing friend, and we are looking forward to having her talk. And so just remember that today this is not medical advice, so please consult your physician. This will be recorded. It will be made publicly available. So if you don't want your name shown, please, remove it from the Zoom or turn your camera off, or if necessary, you can exit this call as well. And then also just remember that we're a nonprofit. So we are funded through donations solely. And so if you feel inclined, please go to our website. There's a donate button at the top of our homepage, and we would appreciate any donations that you may have. So with that, I'm going to turn it over to Ida, who is going to tell us about her research in TNF. Ida, I'll let you take it away.
Brian, thanks very much. So I do want to say that I kind of focus more on the patient group here because I figure that if we want to get deeper into science, we can just connect with the researchers here, and just wanted to bring everybody on the same page and kind of think what does the research we're doing, or specifically in my team, and how we can think about it in terms of translational approaches and how we can take it into patients. So the title, which was kind of discussed with Brad, actually focuses on TNF. And just want to make sure that that's my presentation. Can you see it?
Yes. If you can go into presentation mode, I think we'll be able to see it. Yeah. Good. This isn't your first rodeo.
Yay! How nice.
We do see the next slide. That's okay. It's your speaker notes.
Oh, we don't want that. So let me.
Okay. I'm gonna stop share and then we'll reshare.
Yeah.
And then we'll do this one. I think that's right. Right?
Yeah.
Yeah.
Is that better for you? Yeah.
Perfect.
Perfect. Yep. Okay. And all right. So the presentation title is how a particular protein, in this case, tumor necrosis factor, might control the immune response in prostate cancer. So, okay.
Let's go to the other side.
Just a second. We'll just organize ourselves around this. So I will actually start with the hypothesis, because we'll repeat the hypothesis several times as we get to know a little bit more about the data. And as you can see, it's clearly stated, we think that blocking TNF with existing approved FDA drugs may enhance immunotherapy efficacy in prostate cancer by reprogramming the tumor microenvironment to promote a more robust anti-tumor immune response. And we really are thinking about checkpoint inhibitors, which are widely available and FDA approved. And this really comes from the clearly unmet need. We are wanting to understand how can we improve the efficacy of checkpoint inhibitors in prostate cancer. And we try to understand what makes prostate cancer cold. So the hypothesis is really to address this unmet need. So just to bring everybody on the same page by refreshing our understanding of TNF's function in inflammation. Inflammatory processes are complex, and TNF plays multiple roles within them. It acts on various pathways, initiating and regulating the body's response. So let's explore these effects in more detail. And it turns out that TNF has been implicated in just about all effects, including in the TNF, in the inflammatory response, but TNF appears to be sort of at the head of the process. And it initiates a lot of a signaling that eventually becomes kind of chain effect and accomplishes what needs to be done to address pathogens or any sort of cell problems that need to be removed from the system. So that includes initiating the inflammatory response, just like I said. What's interesting is it's heavily involved in activating endothelial cells, helping the peripheral immune cells reach their targeted tissue sites. So it helps to localize the immune cells to the actual tissue where pathogens took over, or there is any other reason to remove these cells in those tissues. So it also increases vascular permeability, further enabling the reach of peripheral immune cells into the affected areas. It turns out that TNF may also be involved in clearing cancer cells, and that's another effect that TNF has. And its role, and then TNF is overexpressed for a period of time. It often leads to autoimmune diseases. And that's kind of a place where TNF has been targeted successfully. And we know today that there are many TNF inhibitors which are widely used in clinic in autoimmune diseases. So Humira is one of the key biologics that are used widely. But the first approvals came in as early as 1998. And obviously, these are widely used drugs. They capture a huge market share, 13 billion globally. And today, we already have biosimilars coming along, such as Amjevita and Sotezo. And so access to these molecules, or access to these drugs, is pretty, it's pretty well addressed. And what we know from using those drugs, or for those drugs being in the clinic from 1998, is that they have a well-established safety profile, and they're considered to be generally safe. While there are some risks, this risk can be managed by effective and can be managed with regular monitoring. So what's interesting to me, and where I'm focusing, is that it turns out that at some point the inflammatory response needs to be resolved to allow tissue healing. And today it's a very popular area of field of research, and the mechanisms driving this transition from inflammation to healing are being well studied. However, what's really interesting to me is that TNF may play a role in this process too. And so TNF has a dual function, and it has an inflammatory function that's very well understood, but it may also have an immunosuppressive function that is less understood. But lately I see more and more literature on that and more research on that. So we are thinking, based on our genomic study, is that in prostate cancer, the prostate cancer exploits TNF, or hijacks TNF-driven immunosuppressive mechanisms, and that contributes to cold tumor microenvironment and reduced response to checkpoint inhibitors. And from there, we are again restating our hypothesis that perhaps blocking TNF, and in this case, blocking the immunosuppressive function of TNF with FDA-approved TNF inhibitors, will enhance checkpoint efficacy in prostate cancer, again by reprogramming the tumor microenvironment to promote a robust anti-tumor response. So how did we come up with all this? And that actually stems from a bioinformatics study of prostate cancer genomics, and we focused on RNA-seq genomics. And when we started this, we were completely agnostic. We wanted to know what happens with the inflammatory microenvironment in prostate cancer, because there are many publications suggested that inflammation plays a role, but there isn't really a consensus of what happens, how it contributes to the prostate cancer. And so we dove into this area. So we looked at the RNA-seq data from prostate cancer patients in our, we approached the cohort that we have at the Morris Cancer Center. So this data comes from Tempus. So while Tempus provides the reports to the physicians, they also upload the raw data from the sequencing of the pathology samples. So all this data is really patient data. So, and I trust patient data more than experimental models in prostate cancer, especially because they happen to be rather weak. So this data is directly from prostate cancer patients. So what we did is to really look at the inflammatory landscape. We binarized our question, and basically what we said is, what is the difference between localized and metastatic disease? So we could see the progression of disease and how the inflammatory proteins are contributing and how they change with the disease progression. And what was really interesting to me was that then we had a look at TNF, and in this case we had 49 patient samples. Then we looked at TNF, we saw that TNF is consistently expressed in prostate cancer with some kind of increase, or it wasn't that high, but there's an increase with progression. So that was kind of interesting that TNF is there and what does it do? But what became even more interesting to me is as disease progresses, the IL-6, which is a cytokine that is activated by TNF or NF-kappa B and other promoter areas that are controlled by TNF, seems to be regressing. And that was kind of unusual to me. I used to be more involved in immunology studies, and my kind of, sort of the dogma I had in my mind is that when TNF high, IL-6 is high. So when I saw this reverse relationship, I just was wondering if it's an artifact or what does it do? And so I pursued that. And we looked at, different, we looked at patient charts to see if there is something specific about this, maybe steroids, etc. But we couldn't detect the connection between treatment and the IL-6 expression. And so we decided to focus specific on the bioinformatics data and pursue it. So in the A panel, you see the heat map, and you can see that IL-6 is highly expressed in localized disease, but seems to regress in metastatic disease, while in TNF, it kind of increases with disease progression, just like I said. We also did box plots, and you can see that there is a high statistical significance and the differentiation between IL-6 levels and TNF levels with disease progression in prostate cancer. So then we had a look at other cytokines that are usually regulated, activated by TNF, and one of them that stood out was E-selectin. So again, you see a similar sort of distribution of E-selectin with disease progression. It just seems to disappear in metastatic disease. However, it's expressed in the localized disease. It's very similar to IL-6. And here we have sort of underneath that, we have a TNF expression. And the box plot also just of E-selectin shows a clear statistical significance in differentiating the levels of expression of E-selectin with disease progression. So then we had a look also at TNF correlation with checkpoint inhibitors, and we did a Pierce analysis. And what's interesting is we saw that indeed there is a strong relationship between TNF and expression of checkpoint inhibitors. And this study actually was done by Caris. So they used an extensive amount of patients. So this were thousands of patients. And definitely there is a statistically significant effect of correlative effect between TNF expression and a checkpoint and checkpoint expression. So what it tells me is probably the cold environment that we see in the prostate cancer is not due to the lack of checkpoint inhibitor, of checkpoint expression. So something else is going on. The checkpoints are there, but somehow they're not being engaged to deliver efficacy. So then we used algorithms to look at the immunosuppressive immune cells, and that was actually published in the paper that we did 2 years ago. And you can see that we were able to detect the disappearance of M1, so of M1 macrophages that drive inflammation, and a higher level of M2 macrophages just by doing a CyberSort TX. So recently I connected with Caris and worked with Andrew. And this is the table that Andrew generated based on Caris data. Now here we're talking about 5,000 patients as opposed to 49 patients that we basically analyzed in our paper. And it looks that Andrew's results are rather consistent with ours, showing that prostate cancer indeed shows in these samples there is a reduction of the immune cell subgroup that actually should be more should be helpful with checkpoint inhibitors. And probably the biggest differential here we see with Tregs in disease progression. So in later stages of disease, we have a 5.5 point increase in Tregs. But we do see a decrease in M2, in neutrophils, in B cells, NK cells, etc., etc. So it kind of like follows our findings nicely. So I want to go back to E-selectin because I'm really interested in E-selectin and what role it plays. So just to remind you again that E-selectin seems to be disappearing, or the level of the expression of E-selectin based on RNA-seq analysis seems to be highly reduced with disease progression. And so we kind of dove in to try to find an association between E-selectin and so its functional activity in prostate cancer. So just to remind you, E-selectin is an interesting molecule and kind of a real good molecule to study because E-selectin is very specific for endothelial cells. So it's being expressed on the vasculature, and we're looking specifically at the vasculature within the tumor, within the prostate cancer tumor. And E-selectin is activated mainly by TNF as well as IL-1B. So it kind of gives us a really good picture of the play of TNF in this case. So the next slide is kind of interesting because it took me only 1 year to get this data. So I apologize for it taking so long. But I was trying to understand how to explore the E-selectin expression in the tissue, looking at actually proteins, because we saw RNA-seq data, but does RNA-seq translate into protein data, which is what we really want to understand. And I was trying to understand what is the system. I didn't want to go into mouse experimental systems because our data is from humans and I wanted to reproduce it in humans. So I came up with this idea. I learned that prostate cancer in some patients often in pathology shows up as a couple of different comorbidities, and one of them, among them, that the patient may have a tissue spot that is BPH, so it's kind of more an inflammatory environment, a benign hyperplasia, as well as a tumor right next to it, and the normal tissue, which is the stroma. And so we went to the pathologist and asked to identify patients who have these type of pathology findings. And then we send it to WashU to stain for E-selectin expression. And what we wanted to see is there a differential of within E-selectin between those three tissues in the same patient. So by this, we kind of reduce the heterogeneity of the data. So this is one patient and governed by one set of genomics. And that's what we pursued. And indeed, here you see on the left that we see a significant reduction in E-selectin protein in the tumor tissue. So the normal tissue has the most selectin protein, BPH has slight reduction, and then in tumor you see a rather significant reduction in the protein E-selectin, which we know is usually associated with endothelial cells, and in this case we think it's associated with a macrovasculature within the tumor tissue. So then we went back to the carriers data to try to understand if there are correlatives that suggest that E-selectin may be somewhat correlated with overall survival response in patients. And so Andrew, who is working with us, produced this data. And what it shows that a high expression of selectin is associated with significant outcomes in overall survival. So that data is encouraging, of course, just correlative data, but it looks like the highest quartile, the patients who express the highest level, which is the quartile 4, in this case, it's the red blood, shows a rather, significant improvement in overall survival. So then we looked at TNF, and somewhat was consistent and encouraging that the less TNF, the better. So while it's not such a clear isolation as with E-selectin expression, it still is consistent, or at least it's consistent with our hypothesis that maybe TNF in prostate cancer is playing an immunosuppressive role. And therefore, the less of it we see, the better it is in terms of overall survival. So Q1 is the quartile of patients that have the lowest expression of TNF. So then Andrew combined the sort of patients who had, or Andrew looked at the dual expression of selectin and TNF, and he found, the data shows that high selectin, patients who express high selectin and low TNF, they show the best overall survival outcomes. So,
Ida, just to do a quick time check,
yes.
how much longer do you have? I want to make sure that we've got room for Q&A.
Yeah, almost done.
Okay, great.
Yeah, almost done. I'll go for that very, very fast. So,
Okay.
so what I want to say that then we looked into literature and we wanted to see if TNF inhibitors were used in combination with checkpoint inhibitors, just to see what has been done in clinic with this particular combination. And interestingly enough, there is a study that's called TISML, and the design of the study was to include TNF. And the reason for including TNF with checkpoint inhibitors was to reduce the adverse effects from checkpoint inhibitors. And then the data was analyzed. The group that had TNF inhibitors in combination with checkpoints showed better efficacy. although it's a very small cohort of patients, but that was kind of encouraging. And also it was found in other studies that TNF blockers reduce the immunotherapy adverse effects, but they do not compromise the efficacy of checkpoints. And then there is sort of emerging data suggesting that TNF inhibitors may prevent secondary checkpoint expression. So, and that's again, this comes from the clinical studies. So, so here's the point of our presentation and our work. And the goal is, could we consider an exploratory clinical study to test our hypothesis? And we're thinking that the approach could be using TNF inhibitors that have a good safety safety profile with checkpoint inhibitors, and conducting this prior to biopsies, and then do post-treatment analysis of biomarkers of immunosuppression, such as the biomarkers which are the proteins we identified, as well as specific immune cells that are associated with immunosuppression. So that's really all the presentation I had for you today, and I'd really appreciate questions and ideas you may have.
Awesome. Thank you, Ida. So if you have a question, please use the raise hand function in Zoom and I'll call on you. There were a couple questions that came in through chat. And Rob Owen, if you are available, I'd love to have you tee these up. One is regarding zinc, and then the other is regarding cimetidine. You want to elaborate on those?
Sure. I was curious if during your research, whether you look at the effect that zinc has on epithelial cells and how it protects them from apoptosis due to TNF. Has that ever been a consideration?
So Rob, in my case, I would need to find the patients who receive zinc and look at their genomics. So I think it would be a really interesting study, but I have not done that.
Okay. I'm finding effectiveness with it. I assist cancer patients worldwide with some of these methods I'm sharing with you right now. How we're able to control TNF with cimetidine, control its production, throw in a couple of other, basically we're regulating neurotransmitters along with doing portions to enhance the immune system through diet and supplements with great success. And I just wanted to see if any of this had crossed your mind. I know you're very focused on the components you're working on, but these may be something interesting for you to consider.
Thank you. I think it's very interesting because when we did our genomics, one of the striking findings was we saw a real significant elevation of GABA expression. And we really couldn't, drive it further because it's kind of complicated, but we were very curious about that. Clearly, there is something going on with the
Oh, absolutely. And it influences P13K, AKT, which regulates VEGF and transforming growth factor beta 1. You put the combination of these together with cimetidine, it enhances the effects of the percorprazine and the alprazolam to really very effectively manage those and several other signaling pathways and growth factors. So I think it'd be something that, if you have time or the desire, be worth looking into.
Rob, maybe just explain to Ida a little bit about your background.
Yeah, I'm sorry. Yeah.
I think we already set you up with Saul Priceman from Saul's presentation.
Yeah. I got a meeting with him here in a couple of weeks. I appreciate that. So I'm a little over a year ago, I was diagnosed with stage 4 head and neck squamous cell carcinoma. I figured out in the middle of my treatment a better way to treat cancer. My tumor resolved in 2 weeks. I followed most of this protocol, except I was not taking the cimetidine. In my case, I had percorprazine and alprazolam. I had a very limited diet supplement regimen. When my doctors couldn't explain why I healed so quickly, I decided to reverse engineer it and figure it out myself. And by doing so, I was able to stumble basically across these things. And I've been building upon that knowledge across all types of cancers. We're having significant success with glioblastomas, with stage 4 non-small cell lung cancers. Just had a stage 3 cervical cancer with mets to lymph nodes, have 70% regression of the tumor in 3.5 weeks with standard of care. I ended my treatment after 3 weeks. It was supposed to be a 7-week treatment. I'm still NED, pretty healthy besides the cold. And so I've been working with integrative oncologists worldwide. I'm working with the University of Utah School of Medicine. Chris Gregg and I are working through this together. I got a meeting with Saul that Brian was kind enough to introduce me with USC shortly. So I work with an integrative oncology group in India where we're treating patients with these methods. I've talked to Dr. Michael Castro. He's in agreement on some of the research that I've found. Basically, I've been looking at it from micronutrient deficiencies driving all DNA mutations. And then by managing it through diet and supplements on that side, you build the immune system up to help fight your cancer cells. And then by managing the neurotransmitter side, the signaling pathways with commonly prescribed medicines that are mostly given to cancer patients, the antiemetics groups, you get the correct combination of those based on gender, pre-existing conditions, the specific type of signaling pathways that the cancer uses to grow. When you put that combination together, we're having remarkable success in managing cancer better.
Thanks for the background, Rob. Really amazing stories, Ida. We can certainly take that offline. By the way, you mentioned Dr. Michael Castro, who also is at CellWorks. I did reach out to him because I know that he has an interest in this. Unfortunately, he, I believe, is in clinic today, so he couldn't make it. But Ida will probably share this afterwards, and he might be somebody else that you might be interested in speaking to. He's a very talented physician. Yeah. Brad, you've got your hand up.
Yeah, Ida, you're focusing on the immune system, let's say dynamics. It's almost like an ecosystem, and there's accelerators and brakes, and you're trying to kind of tune the immune system, I think, to work effectively in ways that would help the patient. Checkpoint inhibitors are the most well-known immunotherapy, I guess, the one with the most experience. But there are also personalized neoantigen vaccines. There are also CAR T and other CAR NK. There are other things on the horizon. There are other immunotherapies that would benefit from this. And just to share a personal experience, I had CAR T 2 weeks ago, and I went through the whole cytokine release syndrome. I had to be hospitalized. It's normally done in the hospital. I was outpatient, but had to be hospitalized when I had the response that I did. So I'm just sort of curious. There's the cold, hot tumor thing, mostly focused around checkpoint inhibitors, but you could also be, say, modulating somehow the immune system generally. Sometimes you want to turn it up, sometimes you want to turn it down. So in my case, when I started having fevers, they would then do things to kind of turn down the fever. But in general, they didn't want me to be doing anything that would modify the inflammatory response. They want the CAR T to run. Mm-hmm. And they want the near fever response, but not the over fever response. So it seems like being able to modulate the intensity of the immune response, or to channel the immune response, to tune the immune response, would be generally a great thing for any immunotherapy, not just checkpoint inhibitors.
So it's interesting because I'm not aware of studies where they use TNF the way done like a rigorous study where they showed that yes, indeed, that's the anticipation, is that you want the inflammatory response, that the inflammatory response is helping to get rid of the tumor. So these are connected. But the surprise was then the rigorous study was done. It was shown that TNF or blocking TNF was not really in any way. So you can decouple the blocking of TNF and the efficacy of checkpoint inhibitors without really affecting the efficacy. So in this case, I'm aware of a study that has some rigor. some scientific rigor, as opposed to just, oh yeah, we want the inflammation to be all out, which creates a lot of adverse effects. So I think it's to be studied with CAR Ts. It's probably a little bit more difficult to do than with checkpoints, because CAR Ts are a little bit more individualized, and these are more anytime you can use one reagent versus you can use a drug versus cellular therapy, it's much easier. So I'm just aware of this data, which I find very interesting and sort of not intuitive.
And I thank you for that. That took care of that question. Just if I could add one more, Brian.
Yeah.
I haven't had a chance to really go through the conversation with Saul Priceman from last week. What would you see at the intersection of Ida's research and his research? Because he was looking at bispecifics and CAR T, and again, the sort of the progress of immunotherapies generally in prostate cancer. And so, how might you characterize the intersection of their research?
I think at like a really high level, it's just complementary. So, what Ida would do with the TNF strategy would simply complement what he's doing from a CAR T perspective. I get these confused. There's a autologous CAR T, and then there's the what's the other one? Ida, help me. Somebody help me.
Allogeneic or
Allogeneic, yeah, allogeneic, right. And so, I know that he's pursuing both of those. He's also looking at multiple targets. So we talked about, rather than just going after, for example, a PSMA targeted CAR T, going after several different targets, just given the heterogeneity of the cancer. That's sort of where his research is going. But I think, what Ida's doing just seems to be completely complementary to what he would be doing. So maybe there's an
does this
opportunity.
change the paradigm at all? I know Rick Stanton, our dear colleague and friend, educated us about hot and cold tumors. Does this understanding of TNF at all modify that simple hot-cold distinction?
That's a real excellent question, Brad. So we're actually now looking at what are the immune cells that, that particular experimental system we designed, where we can look in the same patient and look at differential expression or differential presence of immune cells that are associated with immunosuppression, so cold versus hot tumor, right? And we're able now to see what are the cells that show up in tumor versus BPH that's considered to be hot, or it has this inflammatory factors that kind of associate with good response to checkpoints, and the stroma. And so that data is kind of trickling in, and it's really interesting data. I'm just really thrilled by it, but it's just too early to speak about it, so we'll write it up soon. But the real issue here is, can we connect TNF to all this? Because obviously there are many different factors. We like TNF because it can be easily targeted, and it has a good safety profile. So we have a little bit of a sort of, hope that TNF will come through on this, but we keep researching this. And I saw a real interesting question. Somebody asked if we could add selectin to the mix. I don't know who asked that, but I saw that question popping up.
Yeah, Rick Davis did. Can you supplement increase selectin in a clinical trial with adding selectin to the TNF inhibitor be a second variable to the test? What led you to selectin?
Yeah, so that's an incredibly interesting question. So I do want to say that we showed in our data shows that IL-6 just seems to be disappearing from the tumor tissue with disease progression, and so does E-selectin. However, IL-6 in the past was considered to be a target in prostate cancer. It was even taken to the point where it was considered to be a driver in prostate cancer, and it led up to a phase 3 study. So what does our data tell us? Is it an artifact, or, what are we dealing with? We discovered, and we actually found some validation through our people's research, is that IL-6 is highly prevalent in the periphery. So the peripheral blood has loads of IL-6, but it doesn't trickle into the tissue. So the tissue seems to, the tumor tissue is very, how should I say, stingy on IL-6 expression. But IL-6 is just all over the place in the blood. And the same is true for E-selectin. So the trick here is that E-selectin needs to be enriched within the tissue or the tumor, because that's where it really plays the role. The tumor also acts like, hey, you know, there is a problem, but it's not here, so go away. And E-selectin really drives the immune cells to get into that localized tissue where there is a problem. So now we don't have E-selectin. So T cells, B cells, etc., the important cells that engage the checkpoint inhibitors. The checkpoint inhibitors are there, they're highly expressed. So that's not an issue. They should work, right? We should be able to knock them out. But if we don't have the immune cells that knock them out, we really didn't do anything. So if we lose the T cells and the B cells that actually present the antigens or present the tumor antigens, and we reduce the checkpoint influence that creates immune suppressive environment, we really haven't accomplished the goal here. We need the immune cells to be highly infiltrating the tumor. So the tumor has a lot of immune cells, but I suspect that they're mostly resident cells that are already trained by the tumor just to comply with the tumor biology and basically helping the tumor. While the peripheral cells that can be trained to recognize the tumor antigens just don't properly infiltrate into this tissue. And so selectin is being looked at. There are a lot of publications that say E-selectin may be driving cancer, so let's knock it out. But I suspect that what is reported is the peripheral E-selectin level, not the localized, tumor localized E-selectin level. So what we need is an agonist, but we need that agonist to be specifically expressed in the tumor. I hope I didn't confuse you too much.
Rick, do you have any other follow-up questions with that?
Here I am.
There you are.
Here I am. So is it possible to supplement the selectin? Because I heard you mention right at the end that you're looking at maybe selectin agonists. Is it possible? And if you were to create a trial, could you include the selectin? Would it be worthwhile in the trial, including this agonist?
That's a real good question, Rick. I think about it every day. I know that, for example, people use TLR agonists because TLR agonists kind of help with this process. But what I'm focusing on in this case is I'm saying that TNF is a direct activator of E-selectin. It's really TNF up, selectin is up. But that's when TNF does its inflammatory act. And I'm saying that in the prostate cancer, TNF ends up being immunosuppressive. So what if I take it out and restart the system? And hopefully now I turn the TNF back to its inflammatory role, which should increase selectin expression in the tissue. So that really is the kind of idea, is to reprogram TNF from immunosuppressive into inflammatory effect, because TNF directly activates E-selectin. It's like straight line.
So either this actually hits on your response to Brad and my other question in the chat, which is really confusing me, which is this contradiction in TNF. So I'm thinking, TNF attracts the T cells, so that's great. But at the same time, TNF seems to sustain the cold environment. So I'm like a little lost on this whole, and I'm not a scientist, as you can hear, but it's confusing me.
So, you kind of like, I kind of dug into the literature because it was extremely confusing to me. When I saw this connection between TNF and IL-6, trust me, I didn't sleep nights. I just like, what is happening? This is a real puzzle. And it turns out that a lot of people looked at that. The issue is, the experimental systems. You have to have the right experimental systems, and mice don't, often kind of mislead us or are not high fidelity representation of what happens in humans. But, the cell is such that it kind of repurposes a lot of the same proteins. It just uses them in different ways based on the context. And because we only have like 20,000 genes, you cannot run a body with 20,000 genes. So you end up using these genes and the proteins that they make in ways that are modular. So they can be activating or they can be suppressive. And we know that, that we understand very well. So it turns out that, once you have an inflammation, you have to stop it at some point because the inflammation really is a killing effect, just kill, kill, kill. And we know that, Brad just told us about how, difficult it was to have this inflammatory response. So at some point you have to stop it. And it turns out that you use similar molecules to stop it. So it's all context related. And one of the sort of, surrogates of TNF being an immunosuppressive is low consistent expression. So during inflammatory response, TNF shoots up in the sky and then it disappears. But in the immunosuppressive environment, it just keeps being expressed, but at very low levels. So it's sustained expression of TNF. It's just a sort of a surrogate. That's one of the patterns that were described in the literature. What's really interesting in clinic also is that we see that in some patients who receive TNF to down-regulate their autoimmune response. So you have a lot of TNF, you get autoimmune response, the TNF stays there, you knock it out, and you get rid of autoimmune response, such as Crohn's disease, colitis, psoriasis. But in some patients, you actually use TNF inhibitors to treat Crohn's disease. And these patients come up with psoriasis. All of a sudden, it's like they get another autoimmune disease. And one of the explanations is that we knock out also the immunosuppressive effect of TNF. So we kill the TNF inflammatory effect, but eventually we also kill the immunosuppressive effect, and then you get the inflammation back. So that's one of the explanations why this effect is observed, and one of the arguments that TNF has an immunosuppressive role in humans. And that's what I find from my studies, that in prostate cancer, the immunosuppressive environment hijacks the TNF immunosuppressive effect. And because we have TNF drugs, we can actually start looking into that, and because they have a good safety profile. And my goal is to find a way to do a clinical trial. And we could do a readout, we could do a very quick trial, which would be, applying this combination of drugs and then looking at the biopsy meta-analysis to see if indeed we made a difference.
Great. So thanks, Ida. I want to get through some of these other questions too. So there's a question, actually, one that I had too. Alexander Leiloff asked, what stage, how advanced in prostate cancer should the patient be, or just to state it another way, are heavily pretreated cancer patients, prostate cancer patients, would they respond better to a TNF treatment regimen versus those that are less, pretreated?
Yeah, so obviously I'm not qualified to answer that question. I'm more thinking in terms of how we can address that question. How we can get to a point of it. So therefore, I sort of suggest a neoadjuvant approach, because we have the tissue. Once we remove the tissue, if we have metastatic tissue, it's more difficult to assess. As far as may it have an effect, I couldn't say. I don't have any data to suggest that. But I'm thinking that, when we still have a prostate biopsy tissue, we could pre-treat the patient with safe, rubber-safe drugs, and then do a readout after we do the biopsy to see if there is a change in this particular immune cells, or did we in some way address the immunosuppressive environment, because we know what is the profile of immunosuppressive environment. We know the lack of T cells, B cells, etc., and we also know proteins that are involved. So we could definitely have a look at that.
Even for the 40, I think it was 49 patients that you had in the study, in the cohort, was there any stratification of patients based upon their EMR data, heavily pretreated versus not?
Yeah, so that's a real good question. What's surprising about the data, we also reproduced it in 169 patients. So we did the 49, we looked at 49, and then we were able to identify another cohort of patients, and we did the same analysis, and it was highly reproducible. And what's interesting about it, that irrespective of the treatment, the consistency of these findings was amazing. You could really see how consistent it is. So that's kind of interesting. As far as we can see the change in this particular expression levels of TNF and the pro-inflammatory factors with disease progression. So we do see a change, definitely, but it's much more difficult to interpret.
Okay. I just want to get through some of the other questions here. Saeed, you have your hand up.
Yeah, thank you. Thank you, Ada. Just one question. Is your analysis based on just one data set you got from the other paper?
So now we have several data sets. So as I said, the first data set was 49 patients, and then we had another 3 cohorts, and the data was reproducible. So 49 was the training cohort, and then 169 was a validation cohort. We haven't published it yet. We will. And also,
but your publication is just based on one paper, right? One data set.
Yes, but like I said, we're in the process of writing our second paper, which will be interesting because we're gonna look at IHC, immunohistochemistry, identifying different immune cells in the prostate tissue as compared to BPH and stroma. So that will be our next paper. And indeed, then we looked at 169 patients, all data was reproducible.
Thank you, sir.
Great. Brad, I think you've got your hand up again, or is that a...
Yeah, no, it is up again. Quick question. This is almost a Brian question. One of Brian's favorite questions is, how could diagnostics and tests around these dynamics point to patients that would be more or less likely to respond? And I'm thinking in particular, unfortunately, Michael Hensley's left, but Boston Gene is very good at multiplex immunofluorescence and spatial proteomics and these kinds of analysis of not just the tumor cell, but the environment around the tumor cell. And so curious if there's any thing that is on the horizon or you might do. I know you said with Karis, but kind of just trying to get at the surround of diagnostics that might personalize in some ways the treatment.
Well, Brad, this is like the best question, because my dream, if I could say what I would really like, is to see a large cohort of data of of checkpoint response in relation to E-selectin, in relation to IL-6, IL-1B, etc., TNF. And the data set I have has to do with Caris. And I think, we're dealing like with 90 patients, or 60 patients. And then we look at the quartiles of expression, we just lose any statistical power there. So, if Boston Gene has more data on patients who receive checkpoint inhibitors, and we could correlate that data with the biomarkers we identified, that would be just dream come true. Yeah.
Michael Hensley from Boston Gene reached out to me while you were talking and wants an introduction. So Ida, this could present a great opportunity for you because they've got nothing but data.
That'd be fantastic.
Yeah,
Fantastic.
yeah. Brad, any follow-ups? Are you good? Yeah, good. There's a question from Stephen Barry about would you combine TNF alpha with androgen ablation blockers in your trial? And your trial would be only for primary cancer neoadjuvant setting?
You know, when it comes to AR, it's very complicated. So, but there is some data suggesting that TNF and AR has some kind of interconnection, or there is some kind of connection there. I haven't looked into that. I couldn't answer this question. I'm just limited on my understanding of how these two work together.
Okay. All right, great. I want to be mindful of time. We're at the top of the hour. I see that some folks have to roll off. Ida, do you have a minute or two in case anybody has any other questions?
Please, yeah, of course.
Okay.
And by the
Does
way,
anyone?
the questions are fantastic, really helpful. Thank you.
Yeah, I told you we've got a really smart group here. Yeah, it is. There was a question from Elaine Watkins. Let's see, regarding CTCs. Have you found any relationship between CTCs and TNF?
And TNF, sorry, Len, no. Didn't have a chance to look at that. Yeah, it's a good question though.
Yep.
I'd be curious to know. Yeah.
Okay. Great. Does anybody else have any other questions? I think we covered everything that was in the chat. If we didn't, please raise your hand and I'll call on you. But I think I got everybody here. Yeah, I think so. Well, great. Well, thank you for creating this presentation. I think you said at the outset that this was going to be a patient-oriented one, but I think we got into quite a bit of science today, which was great. And you know, the great thing is that our group here can go into the science, which is awesome. We have a really smart group of people here. So Ida, thank you so much for your time, for educating us. And I'll be making at least one or two introductions after this meeting that will hopefully help you with your research.
Yeah, and Brian, if you want to stop by tomorrow, that will be fantastic.
Yes, well, I'll try to make that happen.
Okay, great.
All right, everyone. Thanks so much.
Thanks. I look forward to hearing from you and best of luck with everything you do.
Thanks so much. All right, take